Hello and welcome to the presentation. The presentation is about the effect of N-acetylation and N-methylation of lysine residue of TAT peptide on its interaction with HIV-1 TAR RNA by Qamer Un-Nisa and Rosner Morrison. The introduction. The post-translational modification of RNA binding proteins regulate many important cellular functions, including nuclear transport, various signaling pathway, and gene expression during stress conditions. Lysine methylation and acetylation in histone proteins are most common studied post-translational modifications to control the expression levels of the related genes. The human immunodeficiency virus contains many regulatory proteins which undergo post-translational modifications to control the rate of transcription, mRNA transport, and other processes. The N-acetylation and M-methylation lysine residues of TAT peptide at different positions, K50 and K51, and the biophysical experiments are performed to calculate the effect of thermodynamics of interaction between TAT peptide and HIV TAR RNA.
Here are the key terms for this paper. Post-translational modification is the expansion of protein function by way of folding, cutting, or the covalent addition of functional groups or proteins. These modifications result in a change in chemical properties or structure. RNA binding proteins are proteins that bind to double or single-stranded RNA. The RBPs influence the structure and interaction of the RNAs and play a major role in their biogenesis, stability, function, and transport. Lysine is an essential amino acid for humans with the chemical formula HO2-CCH-NH2-CH24-NH2. As an essential amino acid, it cannot be synthesized de novo or natively. N-acetylation is the transfer of an acetyl group to nitrogen in amino acids. This can occur through both irreversible and reversible mechanisms. This process requires the enzyme N-acetyltransferase. N-methylation is the transfer of methyl groups to nitrogen atoms in amino acid side chains. The reaction is catalyzed by methyl transferases. The compensation effect describes the behavior of a set of associated chemical reactions which show a kinetic or thermodynamic linear relationship. Enthalpy-entropy compensation is the correlation that can sometimes be seen between the estimates of the enthalpy and entropy of a reaction based on temperature dependence data.
The CD spectroscopy, also known as the circular dichroism, were recorded on a JASCO spectropolarimeter. The cuvette was used for acquiring spectra of 1 cm in length. CD spectra was recorded from 320 nm to 200 nm with the average of 3 accumulations at 25 degrees. The isothermal titration calorimetry was carried out at 25 degrees on microcal VP ITC. The titration of HIV-1 TAR RNA with TAT and different modified peptides were done by injecting 4 microliters aliquots of 350 micromole peptide from a 250 microliters rotating syringe into an isothermal sample chamber containing 1.5 ml HIV-1 TAR RNA solution. The duration of each injection was 8 seconds and the delay between the injections was 180 seconds. The first injection of each titration was used as dummy injection and then was removed from the analysis. Each injection produced a heat burst curve and the area under each curve was determined by integration to obtain the measurement of the heat associated with the injection. The net enthalpy change for HIV-1 TAR RNA stem loop interaction with TAT and different modified peptide were determined by subtraction of the heat of dilution. Table 1 shows that the binding parameters obtained from these bits. The results of the CD spectra as shown in the diagram of TAR RNA were collected in the absence and presence of different modified proteins. In figure 2, the results show that the CD spectrum of RNA in the absence of peptide showed a positive maxima at 267 nm and negative minima at 235 nm minima as well as 210 nm. The minima at 210 nm was more negative compared to 235 nm minima. The diagram shows the CD spectra TAR RNA and the effect of binding of different peptides on the structure of the RNA at 25 degrees. The results of the temperature-dependent UV spectroscopy in Figure 4, it shows that the lysine acetylation 50 has a negative effect on the binding affinity of TAT peptide to TAR RNA and results in 8-fold reduction in binding affinity as compared to the modified TAT peptide binding affinity. As you can see in the results, the surface plasmon resonance, which shows the diagram of the sensogram binding kinetics of TAT peptide along with the fitting curves obtained from fitting using two independent size models. As shown in Figure 5, the analysis of the kinetic sensogram of binding of TAT peptide showed the association rate constant.
Aims of the research. The research aims to determine the thermodynamic and kinetic effects of N-acetylation and monomethylation of lysine on the interaction between HIV-1 TAR RNA and its cognate binder TAT peptide. Earlier research has shown that lysine methylation has been linked to diseases related to the growth and development of an organism. TAT is an important regulatory protein of HIV-1, which can regulate the rate of transcription elongation and aids the propagation of the virus in the host cell. Both in vitro and cell culture-based studies have shown that the acetylation of K50 and K51 of TAT reduces the production of full-length infection variants. However, the monomethylation of K51 by lysine methyltransferase CET7-9 KMT7 results in an increased HIV transcription. The study ultimately aims to increase understanding of what facilitates or inhibits the HIV virus. Limitations of the research and possible future experiments. Arginine also performs a regulatory function at both the DNA and RNA level. The methylation of Rev protein at arginine residue restricts its ability to bind to Rev response element, RRE, and therefore impedes its export from nucleus to cytoplasm. Studying the effect of acetylation and methylation of arginine residue and TAT protein could shed more light on how post-translational modifications affects the functionality of the virus. In conclusion, the research has shown that the effects of N-acetylation and N-methylation of lysine on the binding of TAT peptide to HIV-1 TAR RNA are position-specific. The location of these post-translational modifications has a marked effect on the binding affinity of the peptide to the RNA and therefore on gene expression.